Dear students, we are continuing with the antigen antibody reactions class. Last class we have completed precipitation and agglutination reactions. So in this video we'll be discussing about ELISA basically, and we'll also see what is immunofluorescence test and radioimmunoassays. So coming to ELISA, ELISA is a very important part of this topic, enzyme linked immunosorbent assay. So from the name itself, there's a term enzyme linked. No? So we are using enzyme linked antigen or antibody for detection of the other kind. That is, if you want to detect an antigen, you're going to use an enzyme linked antibody. So enzyme linked antibody is used, and we are going to do it in an immunosorbent platform. That is, you need a material which is immunosorbent, that means which can absorb antigen and antibody, which is absorbing immunogens like antigens and antibodies. So most often we use materials like polystyrene, and we use a micro titer plate for this. You can see the figure here. It's a 96 weld micro titer plate, and this material is immunosorbent material, so which can absorb antigen and antibody to the base of the well of the plates. So that is why this test is called as enzyme linked immunosorbent assay. We are using an immunosorbent material, and we are using enzyme linked antibodies for detection of antigens or antibodies. So there are different types of ELISA. There are basically 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So you should know what happened in each of them. So direct ELISA forms the basic thing. If we understand direct ELISA, it will be easy to understand the rest of the things. So in this figure, you can see direct ELISA, you're going to detect the presence of antigen in patient serum. So we want to find out whether a particular antigen is present in patient serum. For example, consider you want to detect hepatitis B. So we are going to see whether the patient serum contains hepatitis B surface antigen. You call it as HBS antigen. So for that, you're going to add the patient serum to this microtiter wells. That the figure you have seen, no, in a microtiter plate there are different wells. So into the well, you're going to add the patient serum. Suppose the patient serum contain antigens and antibodies, those things will be coated on the well, because this is an immunosorbent material. So if it contains hepatitis B surface antigen, it gets coated on the well, surface of the well. Second thing you're going to add is, you're adding a enzyme conjugated antibody. Enzyme conjugated antibody. So you're going to add an enzyme conjugated antibody which is specific for this particular antigen. So then you wash it. So in between each step we wash it. Why you want to wash in between is, we want to remove all the unbound fractions. We want to retain only the bound things. The free things you want to remove. That's why you are washing in between the steps. So after washing, only the bound fraction will be remaining in the well. All the free antigens, antibodies will be washed away. So if suppose the patient's serum contains that particular antigen, this enzyme conjugated antibody would have bound to it. So next thing what you want to know, we want to visualize it. We want to see whether the reaction has taken place. For that, you want to bring a color to this. In order to bring a color, you're going to add a substrate to which this enzyme will act. So if suppose you are adding enzyme, we are using enzymes like alkaline phosphatase or horseradish peroxidase, you add the corresponding substrate for that. So when the enzyme reacts with its substrate, it will produce a colored product. So that is the final thing that happens in ELISA. Finally, the enzyme reacts with its substrate to form a colored product. So when an antigen-antibody reaction has taken place, finally that enzyme which will be attached to this antibody will be reacting with its substrate, forming a colored product. So you get a color only when antigen-antibody reaction occurs. So that is it. So in this you can see initially there was no color, now there is a yellow color. Why there is a yellow color? This enzyme has reacted with its substrate, releasing the colored product. So this has happened only because this enzyme conjugated antibody is bound to this antigen. Otherwise it would have been washed away. So you have this positive reaction or a positive color in the well indicates that this patient's serum contains that particular antigen or antibody of interest. That is it. So that is the basic ELISA, and that is direct ELISA. So this is a direct ELISA. So now how it is different from indirect ELISA? If you understand direct ELISA, now everything is easy. Only certain things will get added to it. Indirect ELISA, just understand that indirect ELISA is for detection of antibody. So you're going to find out the presence of antibody, and suppose you're going to find out the antibody to HIV. Want to know whether the patient is HIV positive. What you do is you're already coating the well with antigens. So you coat the well with HIV antigens. So we are already coating the well with HIV antigens. Now you are adding the patient serum. Suppose the patient serum contain anti-HIV antibodies, they will bind to these antigens. So here you can see the antibodies have got bound to the antigens present in the well. Now we want to detect it. There is no enzyme or substrate already added. So you're going to add a secondary antibody against the primary antibody. So we have an antibody of interest here. You're going to add a secondary antibody labeled with the enzyme. So here there are two antibodies. Now already the antibody present in the patient's serum has got attached to the antigens in the well. So in order to detect that, you're going to add a secondary antibody which can bind to the primary antibody, and that secondary antibody will be conjugated with an enzyme. Then you add the substrate and you get a color. That color indicates that this particular patient's serum contains the antibody. Why we are sure that if color comes, there is definitely the presence of antigen or antibody? Because we are washing in between the steps. In between the steps, you are washing to remove the unbound antigens and antibodies. So only the bound fraction will be remaining in the well. If you get a positive color, positive reaction, that indicates there is definitely a bound antigen antibody in the well. Okay, so that is indirect ELISA. So indirect ELISA is for detection of antibodies in most of the case. So to detect antibodies, you're coating antigens in the well. We have the patient's serum which contains the antibody, and you are adding a secondary antibody conjugated with the enzyme. That is indirect ELISA. The next one is sandwich ELISA. So sandwich ELISA is interesting. Why? Because you can see in this figure, you can see the antigen is sandwiched between two antibodies. That is why it's called sandwich ELISA. So here most of the time we are going to detect the presence of antigen. So for that, in direct ELISA also you can detect antigen, but it's not that specific. So to make it more specific, what you do is you coat the well with antibodies. Already you coat the well with antibodies. Those antibodies are called as capture antibodies because they are able to capture the antigens. Now you add the patient's serum. Serum contains a particular antigen. This capture antibodies will capture them. Then you wash it, so all the unbound antigens will be removed. Next step, you add the secondary antibody, which is conjugated with the enzyme. That also will bind to this antigen. So you get the antigens sandwiched between two antibodies. One is the capture antibody and the other one is the enzyme conjugated secondary antibody. Finally, you add the substrate. Enzyme reacts with the substrate and gives the color. So finally, your well looks yellow in color or any other color depending upon the reagent. The color product indicates that your serum, the serum which you have added, the test serum contains that particular antigen. So here in sandwich ELISA, you're going to detect antigen. To make it more specific, you're coating the well with the capture antibody. That is it. And last one is competitive ELISA. It may be a little bit confusing, but the only thing is, remember, everything in competitive ELISA is like indirect ELISA. But in competitive ELISA, if color is present in the well, it indicates that the patient's serum doesn't have that particular antigen or antibody. That is it. So how you do that is, first of all, we want to measure antigen. You incubate the reagent antibody and the patient's serum in a test tube or in whatever is available. You incubate both of them together. So what happens is, if the patient's serum contains the antigens, the reagent antibody will bind to these antigens. Imagine these ones are the antigens of interest. So after that, you add this mixture to a well which is already coated with antigens, like the indirect ELISA. So here, you know what happens is, only the free antibodies can bind to these antigens coated on the well. The bound antibodies cannot bind to these antigens because they are already bound. They cannot bind. Now you wash it. Every bound thing, everything which is not coated to the well gets removed. You add the secondary antibody conjugated with enzyme, you add the substrate, you get the color, but this color will depend upon how much antibodies were freely available in this mixture. So that depends on how much antigen was present in the patient's serum. So if the patient's serum contains high amount of antigens, more of antibodies would have been consumed. So very less antibodies would have been available for binding to this ELISA plate. Right, so if the patient's serum contains more antigen, your reaction ELISA may be either negative or it will be very weak color. The color will be very less. If the color intensity is more, that indicates more free antibodies were available. That means the patient's serum doesn't contain much of antigen. So that is it. So basically here there is a competition between the serum antigens present in the patient's serum and the antigens coated on the ELISA plate for this particular antibody. So if this contains more antigen, more antibody would have been used and less will be available for the ELISA test. So here the color intensity is inversely proportional to the amount of antigen present in the patient's serum. So there is a kind of competition. That is why it's called as competitive ELISA. So once more we'll see direct and indirect because I want you to have the concept clear. Competitive ELISA is a bit tough, but even if you omit it, you have to be thorough with direct, indirect and sandwich ELISA. So in direct ELISA, we have an antigen coming and getting coated on the well. You add the primary antibody. Here the primary antibody itself is conjugated with the enzyme. There is no secondary antibody in direct ELISA. Then you give the substrate and the substrate will give a colored product. You get a color reaction. In indirect ELISA, you want to find out the presence of antibody. For that, you're coating the well with an antigen already. You add the serum, so the antibody gets coated onto this antigen, bind to this antigen. You add a secondary antibody conjugated with enzyme and you give a substrate. You get a color. So here there is a primary antibody which is present in the patient's serum and there is the secondary antibody which is conjugated. Suppose you are coating the well with an antibody. You're adding an antigen, then you add a secondary antibody conjugated with an enzyme, then you add the substrate. You call this as sandwich ELISA. So remember, in sandwich ELISA, we have antigen sandwiched between two types of antibodies. One is a capture antibody and the other is the secondary antibody. So that is about ELISA. These are the different types of ELISA. Please be thorough with it. And what are the uses of ELISA? It's used for diagnosis of infectious disease. I told you, for detection of HIV antibodies, for detection of hepatitis B markers, not only antigens, also antibody. Rotavirus, which is a virus which causes diarrhea. So to detect rotavirus antigen in feces. For detection of enterotoxin of E. coli in feces. So for detection of toxins, detection of antigens, antibodies, for all the detection diagnostic purposes, we have ELISA as the major test. So ELISA is the most important test which we use for diagnosis of various diseases. So that is about ELISA. ELISA is an important 5-mark question. We'll see just what is immunofluorescence and what is radioimmunoassay. Immunofluorescence, we have fluorescent dyes. Okay, like fluorescein isothiocyanate, which is a blue-green colored dye, or a lysamin rhodamine, an orange-red colored dye. So fluorescent dyes, when we are using, you need a fluorescent microscope to visualize it. So what does this fluorescent dyes do is they will absorb the UV light and they will emit visible light. So you'll be able to see that greenish or bluish color fluorescence when you see through the microscopes. So what you do is, we have the immunofluorescence test, means we are going to detect antigen or antibodies with the help of a fluorescent dye. So what you do is, here you can see, you take a slide, you take the patient sample and you want to find out whether the patient sample contain antigen A or antigen B. For that, what you're going to do is, you add a primary antibody which is conjugated with the fluorescent dye. Here it's a greenish colored dye. Here it's a reddish colored dye. So if you get a greenish color fluorescence through the microscope, it indicates that the patient's serum contains A antigen. So this is called as direct immunofluorescence. And you have just like what you have seen in ELISA. No, it's a direct, you are using a single antibody conjugated with a dye. Whereas in case of indirect immunofluorescence, what you do is, you take this slide which is already coated with antigens. You're going to find the presence of antibody in the sample. So you have a slide coated with antigens, antigen A and antigen B. You add the serum. If it contains anti-A antibody, then you add the secondary antibody to this anti-A conjugated with a particular dye. If you get a positive fluorescence, that indicates this particular patient's serum contains anti-A antibody. So here you have a primary antibody which is present in the patient's serum, and you add the secondary antibodies conjugated with the fluorescent dyes. That is why it's called as indirect immunofluorescence. It's mostly like your ELISA, but we are conjugating it not with enzyme but with fluorescent dyes. So that is about immunofluorescence. And finally, about radioimmunoassay, you don't have to know much about radioimmunoassay. It has got a wide range of applications in diagnosis of diseases, in detection of hormones, in various biochemical tests and all. What you do here is we use radio-labeled antigens. So you label the antigens with a radioactive substance and you measure the radioactivity. Okay, instead of conjugating with enzymes or fluorescent dyes, we are conjugating with a radio-labeled agent, radio-labeled substance or material or compound. Okay, so here actually it's like a competitive ELISA because we incubate the antibody present in the patient's serum with a radio labeled with we incubate the antibodies, a known antibody with the patient's serum which is supposed to contain a particular antigen, just like in competitive ELISA. So then you add the radio labeled antigen to it. So if this antigen is very less, what happened, all of the radio labeled antigen will come and bind to these antibodies. And so you have a lot of radioactivity. That indicates the patient's serum does not have antigen, much of the antigen of interest. Whereas if this antigen is present in plenty in patient's serum, all of them would have bound to these antibodies and no antibodies will be free to bind to the radio labeled antigen. So you won't get much of a radioactivity. So if you get a higher radioactivity, that indicates the patient's serum doesn't contain much antigen. If you get a lower radioactivity, that indicates the patient's serum contains more of antigen. So it's like a competition between the test antigen and the radio-labeled antigen for a particular amount of antibody. So that is radioimmunoassay. Just remember, we are using radio-labeled antigens and they have a competition between the normal antigens in serum for binding to a particular antibody. So that is radioimmunoassay. So finally, once more, we are seeing the previous year questions. ELISA and agglutination, I repeat, those are the most important topics, very important, repeatedly asked. So please make notes on it and please be thorough with it. Please know the types of ELISA, types of agglutination reaction, and if possible, learn to draw the reactions, ELISA reactions, so that it will be better, easy to convey. And precipitation and immunofluorescence also you should learn, and that can also be asked as a question in exam. That we conclude antigen antibody reactions. Thank you.